Good morning. So first, I want to thank the organizers of the conference for supporting me to be here presenting work from our group, the Coral Assist Lab, on identifying heat-tolerant corals and the implications for reef restoration via selective breeding. So the data that I'm presenting here was produced by the members of our lab, led by Dr. James Guest, with the funding of a 5-year ERC Horizon 2020 grant and the support of Newcastle University and the Palau International Coral Reef Center. So assisted evolution is currently proposed as a management strategy to prepare reefs to future climate change scenarios or to restore areas that have been impacted by marine heat waves. One of the approaches of this framework suggests selectively breeding colonies with high heat tolerance to produce the material to seed or outplant new corals at target areas. But to do this, we first need to identify heat-tolerant colonies, which is something relatively easy to do during a marine heat wave. In this picture that I took back home in Venezuela at Los Roques National Park, we see two Colpophilia natant colonies during the bleaching event of 2010. The colony on the right is completely bleached, and I can tell you that it died later, while the one on the left survived after the heat wave. So during the rest of this presentation, I will be referring to heat tolerance as the likelihood of a coral colony of surviving an individual warming event. So coral heat tolerance, as Carly mentioned yesterday in her talk, is a really complex and multivariate trait. It's complex and multivariate because it's influenced by many genes and environmental factors. Besides, there is no single metric that can summarize the response of the whole holobiont. In adaptation terms, it should consider the survivorship of the colony to a heat stress event, and not only the bleaching response, given that we know that bleached colonies can recover after a temperature stress. However, one of the difficulties associated to characterizing and identifying heat-tolerant colonies is that without the occurrence of a heat wave, emulating temperature stress conditions is difficult, and maintaining corals in aquariums for long times can be challenging with some species. So we developed an experimental methodology to identify individuals with distinctive heat tolerance, which was published at the beginning of last month in Proceedings of the Royal Society of Biology. We characterized the resistance of 102 colonies from a single population to an emulated heat wave. For this, we took 6 fragments per colony and exposed them to stress conditions in aquariums. We used a temperature profile that mimics a natural marine heat wave of 5 weeks in duration and reached almost 10-degree heating weeks, which is similar to heat stress conditions during previous marine heat waves that have occurred at our study site and that resulted in mass coral bleaching in 1999 and in 2010. So every second day, we visually inspect the fragments to assign them a health status score. Either they could be healthy, partially bleached, bleached, had partial mortality, or be dead. And these scores were used to estimate a bleaching and mortality index for each colony based on the proportion of fragments in each health score category. So this was adapted from Tim McClanahan's bleaching and mortality index. And this continuous variable was then used to compare the phenotypic response of colonies throughout the duration of the experiment. Low values in this index indicate low bleaching and mortality, while high values refer to high mortality of the colony. But we also wanted to have a really simple categorical classification for coral heat tolerance. Either they could be high or low intolerance. So we classify low heat tolerant colonies as those where all replicate fragments died by the end of the exposure, while the colonies with all fragments alive at the end of the stress were classified as having a high heat tolerance. According to these categories, we identified that a third of the population having low heat tolerance indicated in this graph with the red color, and another third having high heat tolerance indicated here in blue. And expectedly, this matched quite well with the average PMI value represented in the x-axis of the graph. But interestingly, we didn't find any association between the heat tolerance and the Symbiodiniaceae community, as colonies were all dominated by Cladocopium, previously clade C. So here in this graph, I'm showing you the response of the colonies throughout the experiment. The x-axis represents the degree heating weeks during the experiment, and the y-axis represents the response, the bleaching and the mortality index of the colonies. The two curves represent the change in the BMI of the colonies that were classified either as having low heat tolerance in red and high heat tolerance in blue. And as you can see, these two groups, they separate quite nicely in the response to the heat stress. And all the low tolerant colonies bleached and died at lower doses, while the high colonies were either starting to bleach or were bleached at that same dose, but were still alive. So one important aspect for the success of selective breeding as an assisted evolution intervention is the maintenance of heat tolerance of a colony through time, meaning through years. So for testing this, we exposed 8 colonies to the same long heat stress experiment in 2 different years, in 2018 and in 2019. And based on the results of 2018, we classified our colonies in 2 groups. 4 were having high heat tolerance and 4 had low heat tolerance. We then exposed them again in 2019 to the same heat stress. And in this graph, I'm showing you the result of this. In the x-axis, we have the BMI during the first bleaching experiment in 2018. And in the y-axis, we have the BMI during the second bleaching experiment. And as you can see, the colonies with low heat tolerance here indicated in red, in both years, they had high bleaching and mortality index. While the colonies with high heat tolerance indicated here in blue, we had 3 of them that have similar bleaching and mortality index. But we had 1 that have similar bleaching and mortality index to the ones that we have classified as low during that experiment. So these results are suggesting us that maybe they could change. So it's not something that is constant, the heat tolerance for a colony through their life, and they could change depending on the conditions. We don't know why, but they might change. And with these 8 points, we're seeing that the high ones, they tend to, the low ones, they tend to stay low, while the high ones could potentially go towards a low heat tolerance. It doesn't suggest that low ones will revert to be high, but this is an hypothesis that needs to be tested better with a higher number of replication. So we're aware that conducting these really long-term heat stress experiments is not practical, and it's a really expensive method to identify heat-tolerant colonies, and I don't think this is the way that we should identify colonies for restoring or preparing reefs to future conditions. And therefore, we are really interested in evaluating if short-term exposure could predict the response of colonies to long-term stress conditions similar to marine heat waves. And to test this, we exposed 19 colonies to a short heat stress of 7 days and a long-term 5-week heat stress. Our results indicate that the BMI during the short exposure, represented here in the x-axis, so this is the short and this is the long, they do not correlate. So the BMI of the same colonies during the long-term heat stress is not explained by the BMI of that they had during the short heat stress. And these results do not support the hypothesis that we can use short exposures to identify heat-tolerant colonies during marine heat waves, at least not this population that we're working with. So now I'm going to give a brief peek on the heat tolerance heritability, another factor that we have investigated. And selective breeding for reef restoration relies on the assumption of heat tolerance being heritable. But this is a coral biology aspect that needs much further investigation. So we estimated narrow sense heritability of heat tolerance, and I'm not going to go into the details of what narrow sense heritability is here. But for this, we use colonies that we have identified as having distinctive heat tolerances as parents, and we selectively bred to obtain up to 34 different single crosses that we have reared up to adult stages. We have exposed to heat stress conditions several of these different life stages, but in here, I'm only gonna present results of experiments that we have done on 3-year-old offspring. So we exposed this 3-year-old offspring to a heat stress of 5 weeks in duration that reached 10-degree heating weeks, which was really similar to the one we used to select the parental colonies. And as you can see in this graph, again, the response of these two groups, they separate quite nicely, the two curves. And the ones that were coming from parents with low heat tolerance were the ones dying first in the experiment at the lower doses. We have estimated values for narrow sense heritability, but due to time restrictions, I'm not going to present them here, but if you're interested in that, please come back and look me, and we can discuss this further. So in conclusion, we have shown evidence of high variability in heat tolerance within a single population, which is really promising, as this can be the material to use for selective breeding, unless that the population has a low genetic diversity, which is the case for many coral species in the Caribbean. But in the Pacific, we don't have that problem. So we could potentially use local populations to restore these areas. Sourcing more heat-tolerant colonies from one single population has the potential advantages of reductions in the likelihood of maladaptation to other local environmental variables, the risk of selecting different genetic variants of subspecies, the probability of modifying genetic structures of the native local population, and the introduction of pathogens. Our results also suggest that heat tolerance is considerable, stable through time, but more research is needed to understand how stable this is. And short-term exposure do not seem to predict how coral respond to long temperature stress conditions, which is what is killing them during marine heat waves. And we have evidence of heat tolerance in offspring at their adult stages, which is really promising for designing any restoration programs. So I just wanted to say Sulang, which is thank you in Palau.